Okay, thank you for the introduction, and also Yan Li Sizang for the invitation. It's my honor and pleasure to be able to participate in this meeting from sunny Sydney. And we've been virus-free for the last few weeks, so fingers crossed, and hope to catch up with all of you very soon. And these are my conflict interest declarations. And my presentation will provide an overview of theranotics development in Australia and also at Liverpool Hospital. And I would share some of our experiences using lutetium-based theranotics, including using DOTA and PSMA, and to share some of our practical experiences. And lastly, I'll discuss some of the way forward. Liverpool Hospital is the largest public hospital in Australia, and also the second oldest. And we have 6 hospitals in the district, and we perform over 450,000 studies per year in my division. Liverpool will be the CBD for the new international airport, and also the planned Aerotropolis. And currently, there are 3.3 billion capital redevelopment projects, including adding 3 new PET-CT and PMI, and also the second cyclotron, to develop a health, academic, and innovation precinct. So nuclear medicine only accounts for less than 0.1% of the total healthcare spending in Australia. And the major growth in the last 5 years has been in the area of therapeutics. And despite the limited Medicare funding, there has been significant investment for radioisotope production in Australia, including the nuclear reactor, the OPA reactor in Ansell Leukerside, and also the synchrotron research facility in Melbourne. And there are currently 10 cyclotrons in Australia. There's a growing list of radionuclides of clinical interest and availability in Australia over the last 10 years. In particular, there's interest in the gallium-68 PET diagnostics, and also I-124 copper and yttrium. For the therapeutics, the highlight was in 2014. The Commonwealth government invested $10 million to build a lutetium production plant in Ansell Leukerside in 2014-15. So this diagram provides an illustration of the evolution and timelines of molecular imaging and therapeutic biomarkers in my department over the last 20 years. And for this talk, the interest of it will be in the gallium-68 DOTA and PSMA imaging we introduced in 2012 and 2016. And we also start providing lutetium DOTA therapy in 2013 and lutetium PSMA in 2018. And we installed our cyclotron in 2017, which became TJ license in 2020. And obviously, technology process is important. Ultimately, for radiopharmaceutical and for theranostics development, it's what I call my master chef, is the scientific staff. And we have Peter Yu heading my research and product division. But obviously, I think that's the key driver for developing theranostics service further. And as a previous speaker discussed, cancer biology has changed significantly. When I started PET imaging 20 years ago, my job is just look at, FDG, metastatic disease. It's very easy. But the cancer biology and how we study has changed significantly more from cellular into interaction with the tumor environment. And I think obviously molecular imaging plays an important role. And the approval, the first immunotherapy, FDA approval for melanoma in 2011 was also a significant step. And so molecular imaging biomarker and profiling facilitate changing paradigm towards biologic definition disease. And for nuclear medicine, the theranostics offer a unique opportunity linking diagnostic and therapeutic biomarkers and to provide truly personalized targeted therapy. And this is an early example from 2012. The patient with newly diagnosed pancreatic neuroendocrine tumor and underwent FDG PET scan, which was negative, no evidence of glucose metabolism. But the DOTA scan showed multiple foci increased somatostatin receptor activity at both the primary tumor site and also in the liver metastasis. We then substitute the gallium with lutetium 177, and as you can see, lutetium 177 has a much longer half-life and it's through beta decay, but also 10% through gamma decay, providing dosimetry opportunity. And after 2 cycles of therapy, most of the somatostatin receptor activity has been turned off, showing good tumor response. So there has been an explosion of the number of immunotherapy and biotherapeutics in the last 5 years, mostly driven by patient-centric clinical trials network. And as you can see, the only theranotic agent on the list was the Lutetium. So this was the landmark study published in 2017, multi-center trials from North America and Europe showing the phase 3 trial benefit Lutetium therapy. And the study showed basically the progression-free survival at 20 months for the Lutetium arm was 65%, and is a 55% improvement over the control arm using long-acting octreotide therapy. And Sevlan is not taken off the market completely, but when we start using that in 2003, there was good randomized study from America, Europe, showing progression for survival in lymphoma by over 2 years. So this is a similar level of survival we see in the Lutetium at the moment. And there are many lessons we can learn why this is no longer in use. But I think one of the key factors is engagement oncologists and to integrate the therapy in ongoing and new drug trial. So to develop a theranotic service further at Liverpool Hospital, I think the true potential depends on the clinicians and investigators needing such technology. We must have appropriate facilities, imaging, radiopharmacy, treatment capacities, including day-stay ward and radiation safety conservation. And also, we must have a framework for engagement and collaboration with our clinicians and to be part of the clinical trials network. And the challenges I see are the personnel, equipment, business process, regulatory requirement, including TGA, GMP compliance production, and to have ongoing translational research and knowledge platform. Prostate and breast cancer are most commonly diagnosed cancer in Australia, and they're also the second most common causes of death in Australia. So this is at least the most commonly used molecules for PS7 imaging and therapy in Australia at the moment. And the ones highlighted in red, the 4 agents are the one I use in my department for therapy, for imaging or for therapy. So the PSMA11 and 1007 for imaging, and the INT and 617 for therapy purpose. And 107 has an advantage of central pharmacy, much better activity, capacity, lower urine excretion, but also importantly, it also provides theranotic links to 617. This is an example of a patient with refractory metastatic prostate cancer, and with significantly more intense and discordant PSM uptake compared to FDG, and the maximum ACV was 221. And we also can perform the post-uptake treatment scan and to make sure the treatment is successful, and also for dosimetry calculation. And after 3 cycles, there's significant partial treatment response and complete response based on PSA level and also on PET scan after 6 cycles. And the patient remained in remission at 18 months post-treatment. So one of the recent trend in Australia was the movement towards a multi-center trial, and for nuclear medicine community to form a clinical trials network to engage with other cancer trial network and dementia, and also to get government funding. So this is just currently is an example cyclotron research we're involved with. And as you can see, there's also several lutetium PSMA and lutetium DOTA trial we're involved with at the moment. Yes. And one recent example is a recently completed therapy trial comparing lutetium PSMA versus cabazitaxel. It's a phase 2 randomized trial with 200 patients enrolled. We are part of the 11 sites throughout Australia, and there's a PI for oncology and also for nuclear medicine at each site. And the preliminary results were presented at ASCO early this year, which showed the overall response rate based on 50% reduction in PSA was 66% for the PSMA arm and 37% for the cabazitaxel arm. And the progression-free survival at 12 months was 19% for the PSMA arm and versus 3% for the chemo arm. Interestingly, there was divergent benefit seen after 6 months, favoring PSMA, and the grade 3 or 4 toxicity is less in the PSMA arm compared to cabazitaxel. So this is just some of the example of PSMA research we are involved with in my department. And we currently involve 2 new trials comparing lutetium PSMA at the earlier time points, comparing with enzalutamide and also docetaxel. So to build a clinical capacity for theranostics service, I think the considerations include protection for the patient, for the staff, and also to protect the environment and to meet regulatory requirements. And obviously, we need to build appropriate infrastructure, workforce, and process. And to set this service up, I think there are multi-level safety precautions need to be considered, both internal and external. The patient needs very close nursing supervision because anti-nausea drug sedatives and amino acid infusion, we had to give patient for over 4 hours as part of the treatment to protect the kidneys. And the nurses and obviously the doctors like myself need to be appropriately protected with correct shielding, such as perfect shield for infusion pump, and also with personal dosimeters. For the environment, we need to have appropriate radioactive waste disposal method, including local waste in the hot lab, and also long isotope in the basement. And we have several patients that can have urine contamination, accidents, and we need to have a way to manage all this radioactive waste. For my hospital, we also have 2 12,000-liter delayed tank to high radioactive waste. So this provides a capacity for us to expand the theranostics service further and to meet the EPA regulatory requirements at the same time. So one of the questions is how long do we need a patient for? For my own experience, that at Liverpool Hospital, we had to usually keep the patients for up to about 6 hours post-Lutetium treatment in order for them to drop the dose below the 25 microsievert per hour. That's the limit in Australia for us to discharge the patients. And for PSMA, we also had to keep the patient, but probably for a shorter period, usually for up to about 4 to 5 hours before we can discharge the patient. And when I set up my current department in 2010, we didn't even, theranostics wasn't still a big thing at the time. So for the currently, we treat up to 6 Lutetium patients in one day. And there's obviously economic reason we can get much cheaper Lutetium supply. And we convert part of our inpatient bay into the treatment area and provide the existing shielding, but we also provide the additional mobile shielding, 30 millimeter thick, and to protect the patient, staff, and other family members. The treatment area is also close to nursing bay, doctor reporting room, and the scanning room. So, and obviously to the toilets, that's very important. We collected very detailed dose exposure data over the last few years. And as you can see, most of the dose exposure is quite small, but for the clinician, it can be quite high. So therefore, we must have appropriate protocol and shielding to protect the treating physicians. And in my new hospital under development at the moment, we actually plan to put in a dedicated 6-bed theranostic day-stay ward linking the clinical trials unit. So I think that's also a very important development. And this kind of shielding data, exposure data is very important for us to build a new hospital and new treatment area. And I think one of the key advantages of Lutetium treatment or lutetium-based treatment is the ability for to perform post-treatment scan and to perform dosimetry. I think that's one area we still need a lot of work and to give us more credibility and to develop service further. I remember when I involved in radiotherapy 20 years ago, they were changing from X-ray based to CT based, and that's why they also provide better dosimetry and dose volume histogram. And I think that was an important improvement for radiotherapy service, and something I think we need to develop further as a nuclear medicine community. And in terms of how many time points and when we need to perform the after scan, my experience that we probably only need to do scans at 1, 4, and 24 hours, and most of the trace will be washing out by that time. So therefore, the area on the curve is probably the additional 24 to 40 hours imaging probably doesn't value to provide better dosimetry calculation. And we use 1 hour imaging as a baseline, so that's 100% injector activity. And we calculate the percentage injector activity in critical organs over the different time period, and with the Olinda program to calculate the dosimetry. As for the Lutetium treatment, the critical organs are kidneys, dosimetry of the kidneys and bone marrow. And so therefore, the patient must have adequate renal function and bone marrow function to receive treatment. We use a GF cut of 40 and protect patient with an amino acid infusion. This is lower than the one published on the North America. They use a cover 50. But for this cover, I think most of the time is probably okay. Depends on the patients, obviously. And the treatment needs to be well differentiated. And most of our patients, they have extensive liver metastasis and carcinoma symptoms. So therefore, we have to give the patient a large cocktail of anti-nausea drugs as part of the treatment. I found one medicine is pretty useful is aprepitant, is a serotonin, central acting nausea drug. So since we start using this drug in the last few years, and also other hospitals in Australia, the incident nausea vomiting has decreased significantly. In terms of overall side effects, it's not uncommon. The grade 3 and 4 side effects overall for Lutetium treatment is about 41%. And patient can have, alopecia, carcinoma-like symptoms during and after treatment. But most commonly, side effects are nausea, vomiting, and abdominal pain because of liver metastatic disease. It can be severe, up to 7%. So occasionally, I need to admit the patients on the same day, and patients can also be presented to the ED afterwards. I will get a call from the ED physician and say, look, you give the treatment, what do you want to do? So this is also poses a new challenge for the nuclear medicine community and for my registrars. And we must be able to manage these patients and have a process in place with the medical oncologists. And amino acid infusion, the kidney failure is rare these days. Most of the marrow toxicity it's only mild, and most common toxicity I see is lymphopenia. Sometimes it can last for 8 to 12 weeks, and we might have to delay treatment because of that. This is an example of a patient with locally advanced pancreatic NET with invasion duodenum and has recurrent bleeding despite palliative external beam radiotherapy. And patient also had chronic renal failure with diabetes and sepsis in the past. So we get the patient Lutetium, 2 cycles, and the benefit, even though it has not gone, the significant benefit increase in terms of decreasing abdominal pain and bleeding. However, the GFR has dropped from 45 to 50, and that's borderline. And so in this case, we have to look at the renal dose from the Lutetium. So we look at 9.2 gray for right kidney, and left kidney is 8.3 gray. And the tolerance dose is 18 gray. And then we also had to look at the renal dose from radiotherapy. So you can see 8 plus 4 is 12, so it's still below the tolerance dose. So the MDT decision was to procure the 3rd cycle. So this is a case example showing, obviously, dosimetry can be very important and need to be used in the right context and in the multidisciplinary meeting setting. And for the PSMA, the critical organs are kidneys. The dose from our own experience from the Lutetium-25 is less compared to Lute. It's probably only about half. And despite not giving amino acid infusion, and marrow toxicity is higher, obviously because of extensive bone metastatic disease. And we normally only give steroids and granisetron for anti-nausea medication. So it's a lot more simplified kind of treatment regimen. And the unique thing to PSMA is a high dose to the salivary and lacrimal glands. So dry eyes and dry mouth, usually just a mild symptoms can occur in up to about 60% of the patients. So looking at the challenges ahead for radiopharmaceutical and theranostics development in Australia, I think the first challenge is that the isolate production capacity is usually centralized, capital intensive, time sensitive, and we need to have backup capacity. And the radiopharmacy labeling for the lutetium ligands usually facility based. So the challenges are obviously can we meet the regulatory requirements and to have standardized production to support multi-center trial. And the third challenge is how to translate this into clinical practice to have sustainable business process and funding model. And the opportunities are there's a cohesive working relationship and collaboration between the industry and the hospitals. And there's also a favorable regulatory landscape for early stage trials and R&D. For us, it's a TGA-based process, and we have to go through institutional research committee and research radiation committee. And obviously Australia, there's a good international standing in terms of clinical development led by IIT studies. The consideration is, do we replicate the production facility centrally or to have more facility co-located at the hospital? And this can also help to determine the many targeted molecules and what's the best in class. And there's also balancing between commercial versus clinical academic needs, and to have a GMP and TJ licensed facility for theranotics as well, to get government funding and to engage big pharma. And lastly, there's also the competing clinical versus academic research priorities. We must have a scope and framework for engagement, and collaborative clinical trials network discussed. And lastly is the multi-skill workforce. And these are some of the emerging radioisotopes of interest in Australia. So we're looking at isotopes that can be applied through the spectrum or the imaging and therapeutics applications. So three isotopes of recent interest are the terbium, scandium, and also copper. And this is a recent publication by my colleagues from Peter and Mac, first in human study using copper-6 for custate. And it's long half-life, so they actually show good tumor retention at 24 hours. And there's also theranostics potential with copper-67. So going forward, for my facility, I think that there's more interest for more solid target radioisotopes production. For lutetium theranostics, my scientific research team had many ideas. I think there can be application for bone metastasis disease, breast, including triple negative breast cancer, liver. We got some interesting idea we're looking at the moment, and also for hypoxia agent cyanobacteries, and other theranostics such as breast alpha therapy, which I think is coming soon, and also potentially also to do DOTA. But I think we should do this in the multi-center trade setting as well, because it's potentially more toxic. And hypoxia, theranostics, radioimmunization, and sentinel tracers. And lastly, my radiation colleague, there's a lot of interest toward what they call the abscopal effect. So it means that there's a radiation-induced immunogenicity at remote metastatic sites. So treating oligometastatic disease can actually increase patient survival. And this is one of the landmark study published recently, showing by having radiotherapy to the primary tumor in the prostate can actually increase survival in patients with metastatic prostate cancer. So this is a very important study. And I attended a radiation oncology conference recently. Interesting, there's a lot of discussion about theranostics. Is that going to replace SBRT, so stereotactic body radiotherapy for oligometastatic disease? In fact, there's interest and talk about potential radiation oncologists going to provide the theranostics rather than the nuclear medicine physicians. So I think something that we really, as a specialty, we must be a key driver and must participate in all aspects, clinical services. And we have the experienced workforce and expertise in this area. But if we don't do that, we may only play a secondary role, just as a supplier tracers in the future. So going forward, I think the theranotic landscape is moving forward very quickly and very promising, driven by the demand to provide better therapeutic impact, patient outcome, and to do it cost effectively. So thank you again for your attention. My apology for not being able to be there in person, but I look forward to seeing all of you in the future very soon. And please let me know any contacts or any questions. I'm very happy to collaborate further. Thank you.
Okay, thank you, Professor Lin's excellent talk. Because owing to the time limit, can I just suggest one question? Any?
Thank you, Professor. I'm nuclear medicine physician from National Taiwan University Hospital, and it's my pleasure to the experience you shared. I have two questions here. I saw there are a treatment bay in your unit, and there are 6 beds. So you treat patient one by one or treat them together? The second question is if the patient has been admitted due to side effect or he has complications after treatment, who will be in charge to take care of the patient? Is the nuclear medicine physician or his clinical doctor? Thank you.
For the first question, we normally treat 6 patients sequentially, usually 20 minutes to half an hour apart. The infusion is about 15 minutes, and so we normally do that over probably 3-hour period. And obviously the issue will be the radiation build up. When we have 6 patients, there's a lot more radiation, urine activity. But also there's imaging capacity. We need to scan the patient for up to 4 hours. So the capacity is also limited by the imaging time available afterwards. But we normally treat up to 6 patients. It's a lot cheaper in terms of lutetium supply. So there's a definite economic advantage. And in terms of the patient care, my oncology usually transfer the patient to me for 6 months during the treatment. So I actually do the clinic, do the follow-up. But it also means if they go to ED, they need to, they usually call me because I'm the one providing the treatment. So I have an arrangement with my medical oncology. So we usually meet the patients and look at the patient together. And a lot of time my residents and registrar, especially a lot of time, most of them are radiologists. So they don't have the appropriate skills to look after these patients.
Thank you again, Professor Lin. I think we should close this section. And if our audience have any question or comment, yeah, we can provide the contact email or even line, so you can communicate with Professor Lin. Okay, thank you very much, Professor Peter Lin.
Okay, thank you so much. Good afternoon.